Alexis Perrol, CEO of OSE Immunotherapeutics. So we are a French biotech listed on Euronext in Paris, and we were founded in 2012. The idea for us is to be strong from research up to proof of concept, clinical proof of concept. So we've done a lot of work in identifying the right targets, both in immuno-oncology and in autoimmune disease. We are trying to bring these products forward and then find the right partners in order to help us develop this product. We have two partnerships, one with Boehringer Ingelheim in immuno-oncology and the other one with Servier in autoimmune disease. The idea for us is today we have an ongoing IO phase 3 program which is developed in non-small cell lung cancer post-checkpoint failure. These are a combination of neoepitopes which have been optimized in order to have a strong binding and then activate and prime T cells. We decided and make sure to have 4 programs in clinical development right now and 4 products with a clinical stage status. As I said, most of the financing of the company, because we did not have to raise any capital since the IPO in 2015, comes from the partnerships we have. Altogether, the potential milestone that we could have access to is 1.3 billion euros, both from Boehringer Ingelheim and Servier. And we invest a lot in our research in order to develop new targets, new product, focusing in, for example, myeloid cells in order to tackle the suppressive environment that they can build in immuno-oncology. The strategy we have is clearly to leverage the expertise. We are based in Nantes, which is the western part of France, and it has been a cluster of immunotherapy and transplantation for the past 25 years. So we address immuno-oncology with the eyes from our scientists on transplantation, which give us a kind of edge or differentiation compared to pure oncologists, I would say, and we have expertise in this field. So the strategy we have for immuno-oncology is first trying to prime and activate T cells. So we do that with our neoepitopes. The second part is clearly to try to transform the tumor microenvironment and tackling the suppressive cells or myeloid cells, and we do that with a monoclonal antibody which is targeting SIRP-alpha. And the last part that we announced quite recently is a bispecific platform with the backbone anti-PD-1 and then innovative targets that we think are interesting in order to especially try to sustain the life of exhausted T cells. In the autoimmune disease part, clearly, obviously, the contrary. We try to downregulate T effector cells or to upregulate regulatory T cells. And all the way we have academic partnerships and then pharma partnerships in order to accelerate and make sure that our projects become products and are tested in clinical development as soon as possible, which give us this portfolio. So the first product, Tedopi, is a combination of neoepitopes. They come from 5 different tumor antigens, and they have been optimized in order to have a strong binding to the T cells or to the MHC. The first clinical trial, which is a phase 3, post-checkpoint failure, is now in Europe, in the US, and in Israel, and we should have the first results on the first step of this study on around 100 patients in Q1 2020. We also developed this product combining it with Opdivo in pancreatic cancer, as we believe, like clinicians who are sponsoring the study, the GERCOR group, that priming T cell could help having a better effect on PD-1, which, as you know, have a very low effect in pancreatic indication cancer, trying to, with Tedopi, our product, turn a cold tumor into hot tumor. As I said, we have also a phase 1 which is ongoing with Boehringer Ingelheim, a product that is targeting SIRP-alpha on the SIRP-alpha CD47 pathway. You have a couple of companies, biotechs, targeting CD47. We decided to target SIRP-alpha on the same pathway, and as such, we believe that there is a differentiation here and a synergy with PD-1. I will come back to that a bit later. We have a research collaboration with the Memorial Sloan Kettering. This is a very good recognition of the science, we believe, and of our research team. We work here on solid CAR-T tumors. We have a monoclonal antibody targeting IL-7, and IL-7 is overly expressed in some tumors like mesothelioma or non-small cell lung cancer. As I said, you can see here the new platform that we have developed. So it's a bispecific fusion protein, a backbone anti-PD-1. The anti-PD-1 has been selected because of its very good yield. Manufacturing is an issue for bispecifics, so it's important to try to get a very good yield in the CMC part. And then we put additional targets, which we believe will help us address the exhaustion of T cells, for example. In the autoimmune part, we have a product which has a phase 2 status, which is targeting CD28. So it's a pure antagonist of CD28. And we are now actively looking for a partner in order to continue the development of this product. And the last product in our portfolio is OSE 127, targeting IL-7 receptor, which is partnered with Servier. So coming back to Tedopi, Tedopi has been developed in the US by Alessandro Sete, who is a specialist of algorithm of epitopes, and he modified epitopes from 5 tumor antigens in order to have a strong binding either to the MHC part or to the TCR part, and as such, we showed that in phase 1 and phase 2, we have a strong T-cell response for this product when you find the antigens in the human body. Why do we think there is an interest? You know that one of the main suspected mechanisms of resistance to checkpoint inhibitor is the HLA downregulation. And through this product, we know that we can upregulate the HLA part. So that's why we are testing it in non-small cell lung cancer. And we see that clinicians are happy to recruit patients post-checkpoint failure when you don't have anything else to give to the patients who received a first PD-1 or PD-L1. The only thing you can give them is docetaxel, for example. So with a product like Tedopi, an immunotherapy, and a synergy with PD-1, this is something which is clearly interesting. As I said, the idea was to have neoepitopes based on the MHC1. So this is for HLA-2 positive patients. It covers 45% of the population, and we know that the reduction of MHC expression can be reversed through interferon gamma, which is why we are using Tedopi in that case. How did we develop that? I mean, obviously, it's based on 10 years of R&D. We bought back this asset just before the boom of Immuno-Oncology, which gave us access to these 10 years of R&D. The epitopes, 11,000 epitopes have been selected from 5 tumor antigens, modify them through algorithm in order to have a strong binding and then to break the self-tolerance. Why did we decide to buy back this asset? Is because we saw in a phase 2, which was done in the US, in very advanced patients, most of them were metastatic, had 3 lines or more of treatment in non-small cell lung cancer. And we saw not only a nice survival curve, but clearly a correlation between the response to the epitope and the survival. So the more the patients were responding to the epitope, the longer the survival. We also saw in the survival curve, and you can see it here on the slide, that 25% of the patients who received Tedopi for 2 years were still alive after 4 years. So you have this plateau that you find in the new PD-1 and PD-L1 treatment in immuno-oncology, in immunotherapy, where patients responding then will be long-lasting and long survivors. We are investigating Tedopi in non-small cell lung cancer for patients who received chemotherapy and PD-1 or PD-L1. They can then enter the study. We randomize them compared to docetaxel 2:1. We will assess with the first 100 patients whether we reach a level of survival at 12 months, which has been defined as sufficient by the agencies and by the steering committee of the company, of the trial, sorry. And then if this is strong enough, we can continue to add 200 patients in order to get the survival compared to docetaxel or pemetrexed. We have been able to show initial case reports at AACR. Why do we think that this is interesting to have a multiple neoepitope vaccine when you have resistance to ICI? Obviously, you can increase the tumor-associated antigen presentation. You can increase the priming and activation of T cells. And that's why we presented during the last AACR some clinicians who are participating to our phase 3 study wanted to show that for patients who had received anti-PD-1 in second line, so the patients were in third line in non-small cell lung cancer, and you can see here that you have one partial response, two long stable disease. The interesting part is that the overall survival after the Tedopi initiation was around 2 years or more than 21 months at least. So these are only case reports. There were no comparison to the control arm and so on, but just to show that we see some early signs of efficacy for this product. As I said, we also believe that priming T cells, activating T cells in cold tumor could be interesting and demonstrate some synergy with the PD-1. That's why we are starting a phase 2, combining Tedopi with Opdivo. This is sponsored by a group of collaborative clinicians called GERCOR. We will assess in 3 arms for a maximum of 150 patients, Tedopi plus Opdivo, or FOLFIRI, which is the standard of care after FOLFIRINOX. Patients will receive FOLFIRINOX for 4 months, then you have to stop because of the toxicity, and we'll see whether, in particular, Tedopi arm or Tedopi plus Opdivo arm are doing better than the FOLFIRI alone arm. The other strategy we are addressing in Immuno-Oncology is clearly to target myeloid cells and macrophages. So we have with this product targeting SIRP-alpha, which is licensed to Boehringer Ingelheim, a first-in-class myeloid checkpoint. We are now doing the phase 1 and dosing the first patients in the coming weeks. We will have the results in next year, in 2021, for the first part. Boehringer is paying all the cost, and which gives us obviously some financing for this study. Why did we choose Boehringer Ingelheim? I mean, it's obviously a very good company focused in oncology. They have a global footprint, and what was interesting is that they have their own anti-PD-1. So we will combine our product with their anti-PD-1 and look at both monotherapy and combination in patients with solid tumors in this phase 1 and the expansion phases which are planned from next year. The deal is very interesting for us. We received 15 million upfront last April. We are receiving right now 15 million because we got the approval to start the phase 1 and we are dosing the first patient. And as I said, the potential total milestone of 1.1 billion plus royalties. Why Boehringer was interested in this product? As we are targeting SIRP-alpha, so on the macrophages, and by blocking SIRP-alpha, you transform the M2 pro-tumorigenic cells into and increase the M1. And in addition, we demonstrated that our monoclonal antibody does not bind to human T cells, hence the possible synergy with anti-PD-1. We have some competitors who are targeting CD47. We think that the differentiating point for us is that by targeting SIRP-alpha, you do not block SIRP-gamma that you block when you block CD47. SIRP-gamma is very important for T cell expansion. So we'll see soon when we start the phase 1 whether this differentiation is helping patients and helping T cells expanding more when we block SIRP-alpha only. We think that bispecific are something which will be more and more important in the future. So we have developed a novel platform of bispecific targeting first PD-1 and then innovative targets. So this bispecific platform called BKI or bispecific fusion proteins, they are built on an engineered anti-PD-1 key backbone. We decided to choose this anti-PD-1 because of its very good yield in terms of manufacturing and the synergy you have with the two targets. The first one, which was disclosed recently during a congress, is BKI IL-7, so anti-PD-1 IL-7. We are also targeting other targets like macrophages, and we will soon be able in 2019 and 2020 to present preclinical data for this platform, which is, we believe, very good value creation for the company and the potential for new partnerships in the coming years. In terms of autoimmune disease, as I said, we have this FR104 antagonist of CD28 product, for which we have done the phase 1. And we are now looking at how to continue the development in phase 2. One target and indication could be rheumatoid arthritis, but as you are targeting CD28, which is a very interesting target for immunologists, you can obviously target other indications in the autoimmune space. What we demonstrated in our preclinical package in particular is that by blocking CD28 you get a better result than blocking CTLA4 like abatacept, for example, and we hope to be able to reproduce these results at clinical stage. The other product that we have is IL-7 receptor antagonist that is currently in phase 1. We should have the results of the phase 1 in Q4 2019. We partner this product with Servier. We are going to develop a phase 2 in ulcerative colitis in 2020, and Servier will develop a phase 2 in Sjögren syndrome. And based on these results, they will be able to opt in for the full license. We expect this to be in 2022. As I said, we have shown that by blocking the IL-7 receptor, you have a nice effect, especially for IBD disease. And the deal is also a potential total milestone of 272 million euros plus royalties on sales. The phase 1 is ongoing with 64 healthy volunteers. The results are planned in Q4 2019. So altogether, these two deals represent 1.3 billion euros plus royalty on sales and will help us increase our financial visibility should these two products move forward until 2020 and even more. Obviously, we are also addressing a lot of academic and public partnerships to help us push forward our clinical programs or our new research programs. So in summary, OSE Immunotherapeutics is a company with 4 products with clinical status. We've got 4 clinical trials ongoing. We have been able to finance ourselves since the IPO through the partnerships, which besides the financing part is obviously a very good recognition of the science in our company. We continue to work in research especially in Immuno-Oncology on myeloid and macrophages. We have this new platform of bispecific, which we believe will be of strong interest for pharma companies, and we hope to produce some results on the phase 1 of the OSE 127 this year. At the end of this year, we should have the first step results of Tedopi phase 3 non-small cell cancer in Q1 2020, and the rest from 2020 to 2021. Thank you very much for your attention.